Hi, this is Miss Litton on a minimum day, and 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 of your students are taking it for the team right there.
That's right, I'm Nate. Hi.
Yell your names out, do a shout out.
Nate!
Excellent. Okay, so we are going to review Chapter 3, Cell Structure and Function. Just a couple things I want to address right off the bat. Number one, I did not give you any additional objectives for this chapter, so there are no additional objectives on the quiz. Number two, I want you to remember that you are going to need to know structures of things. So if I show you a diagram on your quiz, you could tell me what that is. So real quickly, let's just review that part. Okay, you tell me what it is. So I'll point to it, you tell me. Okay, what is this right? Hi, I want to be a pin. Oh, I am a pin. What is that right there?
Mitochondria.
Good. What is this?
That's the nucleus.
Nucleus. What's this?
Nucleolus.
What's this?
Vacuole.
What's that?
Chloroplast.
Chloroplast. And this?
Those are for sure
Golgi
apparatus.
Golgi apparatus. What are these?
Smooth ER.
Smooth ER. And this would be?
Rough ER.
Good. What would be this stuff right here?
The cytoskeleton.
Cytoskeleton. And this is the?
Cell wall.
Good. And what happened? Oh, what would be like little dots in here?
Ribosomes.
And these are? Centrioles. Okay? Good job. I think you've hit. And if I had little vesicles like this, you would not know if that little vesicle was a peroxisome or a lysosome or an endocytotic vesicle or a small vacuole. So I'm not going to ask you something that's like that where it could be many different things. I could ask you this one because you obviously know that that's a large central vacuole, right? Okay. The other thing is knowing the parts of the cell theory. All living things are made out of?
Cells.
Okay, and it's the structural and functional unit. Okay, that's what we're made out of cells. It's the structure of things, and it's how those things work. And then you only get cells from other cells. Make sure you know that basic. We talked about the size of cells, and we said cells are basically very, very what? Small. Why are they small? They need a large surface area per volume. Large surface area per volume. The analogy, Sorry. The analogy I would give for that is if this were a large.
It's gonna happen right now.
Okay, that's not what I want. I want this. Okay, no, I don't want that. Go away. I want this. If this were a cell, okay. So I could measure the surface area per the volume. You can see how close. Look at the distance between this edge and this edge, right? And then if I had a cell, though, that was this big, right? Do you see how now I have a larger surface area per volume inside, right? As opposed, look at this cell, right? Even a larger surface area per volume inside of it, right? So that's why you want to have cells that are very, very small, so they have a large surface area per what you have inside of that cell. Okay, so that's why cells are small. Okay, then we started talking about prokaryotic cells. So you want to know the hallmark of prokaryotic cells. What could I find in a prokaryotic cell? Tell me.
Flagella.
DNA. Okay, DNA, and that DNA is located where? In the nucleoid region. Nucleoid region. I don't have a nucleus because I don't have any membrane closed organelles. Hang on one second. I have a student, former student. You?
You can see them, but like they can't see you at all.
What do they mean by large?
So it's really funny Oh my gosh, it's still recording. Hey guys, so you guys are gonna get 100 on this test. There is no additional objective. How hype is that? Woo woo! She's out there hugging the former colleague. No, student. There's flowers. They're still talking. Still going.
Sorry, I'm recording this review right now.
Shh. She doesn't know how to. Shh. Okay, we're back. We're back.
Sorry. I had to go say hi. Okay, so What else do they have?
Cell membrane.
Cell membrane. Okay. What could be outside of the cell membrane?
Moving.
So we have a cell membrane. What goes right outside of the cell membrane? Cell wall. Right? And then what would be outside of the cell wall?
Cytos.
A capsule. Good. Perfect. What else do these cells have? Moving things? What are the moving things called? Yeah, you could have flagella. You could have cilia. What else could you have?
Flagella, cilia, and reproductive.
Ribosomes. What happens on ribosomes?
Protein synthesis.
What? Protein synthesis. Okay. What is the significance of a cell membrane? What does a cell membrane do? It decides what?
Goes in and out of the cell.
What goes in and out of the cell. Now, we have already moved on, right? In chapter 4, we're already talking about this. So this would be really familiar to you now. Okay, but it decides what goes in and what goes out. What are these called right here? Phospholipids. What are these? Proteins. Good. All right. And then this, it's not like you have to spend a bunch of time. If you look right here, we can see nucleoid region. We can see ribosomes. Plasma membrane, so it's membrane, and then it goes cell wall, and then this glycocalyx. We've already learned about glycocalyx, right? Glycolipids and glycoproteins. We know about flagella. What are these fimbriae right here? What do they help do?
Move.
No, cilia would do that.
Oh.
They attach. Fimbriae help you attach. And then the sex pilus is for what it's for. It extends, so you have, and that's the one you were trying to remember, or you remembered, the sex pilus between two bacterium so they can have sex that way. Okay, that's your basics on a prokaryotic cell. And then the next thing we moved into, plasmids, extra rings of DNA. Okay. Then the next thing we kind of talked about is prokaryotic cells versus eukaryotic cells. And eukaryotic cells are larger than prokaryotic cells. Prokaryotic cells are found in two domains, archaea and what else? Bacteria. Archaea and bacteria. And eukaryotic cells, we have these different kingdoms right here, and we've already learned about these already. So focusing on eukaryotic cells, one strategy might be finding a structure, a plant or an animal cell, where they've given the structure and the function of everything that you can do as a review, and I already kind of recommended that you do that, because on the quiz, probably what I'll have you do is I'll say, here's a structure, now tell me what it does. Or can you, and which one of these is wrong? You know, so you want to be really comfortable with that. So if I was going to pick out some different items on here, because I don't think you can see the blue writing, right?
No.
Okay, so if I was going to pick out, let's start with here with the nucleus. What would we say about the nucleus as a function?
It is the brain of the cell.
Brain of the cell. That's a great word for nucleus, brain. What else could we say about it? It contains the DNA. Good. And if I focused on the nucleolus, what's getting made in the nucleolus? Ribosomes are made, right? Okay, so now we're knowing structure and function. So what's right outside of the nucleus? What's this stuff right here? Rough ER. What makes this stuff rough right there?
Ribosomes.
Ribosomes. So if I ask you where are proteins synthesized, you're not going to say on the rough ER. You're going to tell me on the ribosomes. Okay? So the ER, what can happen inside of the ER? If proteins are made, they can move through that ER and be modified within that ER. It could also move over to, this one doesn't have any ribosomes on it. We call it the what?
Smoother.
Yeah, okay. Smoother. I like it. Smooth ER. Smoother. And then lipids are synthesized in there, and carbohydrates are synthesized in there. Okay, what is this right here again?
Golgi
And what do we think of for him?
Golgi body.
Yes. What does he do?
He transports the cell to the cell membrane.
He modifies and packages and kind of puts an address label on wherever this is supposed to go. Because it could be being brought to the cell membrane to be secreted, or it could be a vesicle that you keep within the cell. And the UPS will put that label message on there, that address is what I want to say. Okay, what is this again right here? What are these? Mitochondria. What happens in the mitochondria? Cellular respiration. Cellular respiration makes a bunch of what for us?
ATP.
Good.
Energy.
Energy, good. Lysosome right here. What would we call the lysosome? The what? Stomach of the cell. Now, that can do actually multiple things, right? Remember the 3 different things we said it could do? The stomach of the cell would be where it digests, it hooks up with some endocytotic vesicle and you digest whatever is inside. But there were 2 other functions we said a lysosome could do. Do you remember? Do you remember? Yeah, it could digest other organelles. And then the 3rd thing a lysosome could do is help the cell to commit what?
Suicide. Cellular suicide.
What is that called?
Suicide.
Apoptosis. So a lysosome can digest nutrients that have been brought in. It can digest another organelle or have the cell commit suicide by releasing its digestive enzymes into the interior of the cell. What did we say these are called right here? Centrioles. And are those found in all cells?
Only animal cells.
No, yeah, we're not going to find them in a plant cell.
Yeah, they don't have churros.
Good. All right, and then we talked about peroxisomes breaking down hydrogen peroxide, good, with catalase. All right, so we hit all of those. I feel good about that. We talked about the endomembrane system, okay? And this would be how you have your nucleus and the DNA that's in here, you get a copy of that DNA and that would be mRNA. The mRNA goes out and gets on a ribosome and then a protein is made there. That protein moves in the ER, it could move into the smooth ER and then to the Golgi and then exit the cell or stay within the cell if you need it. That's the whole endomembrane system. When we look at a eukaryotic cell with all of its membrane enclosed organelles in it, okay, how could a eukaryotic cell have evolved from a prokaryotic cell? Do you remember that part? EndoSymbiotic hypothesis.
Do you want me to leave your eggs in your
Oh,
room?
sure.
Okay, and I don't know how they're going to be organized, so you might have more than you need. And I guess
Well, why don't you just keep them in the main fridge and I'll just go get some.
Are you sure?
Yeah,
and
positive.
I thought I'd try to get here early tomorrow to help you and Ellen get the things that you need and get your eggs set up in your
Oh,
room.
okay.
Okay?
Thank you.
All right.
You're awesome.
I'm gonna go
Hashtag awesome
shopping
sauce.
now.
Okay.
My eggs and my vinegar.
There we go. You're setting up eggs and vinegar. 0, don't forget. We got to set up our eggs and vinegar in the morning.
Oh, yes. Dude, that's
Yeah.
like my favorite breakfast.
Okay. Then let's talk about what we didn't really talk about in oh wait, let me go back here. What is this for?
For cellular.
Not for
Oh,
cellular.
for photosynthesis, for energy
Photosynthesis.
for the cell.
What's the large central
Vacuole.
vacuole for?
For storing water for the
so
cell.
it doesn't collapse.
Yes, turgor
And
pressure.
structure.
Okay.
Turgor
Now,
pressure.
cytoskeleton. Remember we talked about 3 sizes of cytoskeleton? The smallest size of all was actin. Then you had intermediate, and then you had the large microtubules. Remember that? And I used pipe cleaners to model that for you. Okay, so you find these 2 things like underneath the cell membrane or around the nuclear membrane in order to provide support throughout the cell. And then the microtubules, we talked about those being like a large cellular railway, that things could be transported on them. And we talked about how vesicles could move along those microtubules.
Oh.
We also talked about the interaction between actin, a microfilament, and myosin, a microtubule in our muscles, called the sliding filament theory. So that has to do with structure and support and movement. When you think about the cytoskeleton, which is the same for us, right? Our skeleton is for structure and support and movement and also protects us, right? Yes?
Is that the dude that's tiny compared to the big thing? Like,
Well, yeah, when it was walking in that video. Exactly. Okay, oh, here, I know that's what I was looking for. EndoSymbiotic hypothesis is how a eukaryotic cell could have evolved from a prokaryotic cell. So the idea is we know endocytosis can happen, that membranes can go out and engulf things. But if a membrane starts to fold in, and it's a really big fold, and that fold loops around the nucleoid region, that could be a way that you form a nucleus. Nucleus, because that is just the DNA encapsulated in a membrane. So that's how you could get the nucleus, that's how you could get the ER. And then the energy organelles, mitochondria and chloroplasts, could have been little animal-like protists or animal-like bacteria and a plant-like autotroph, a little single-celled autotroph, and they went and lived inside of another prokaryotic cell. And our evidence for that we looked at is mitochondria and chloroplasts have their own DNA, and it's circular, just like a bacteria. And they have their own ribosomes. They can make their own proteins. And they're self-replicating. They can make more of themselves. So that is your evidence for the EndoSymbiotic hypothesis. Questions about that? No? Okay. And then that, cytoskeleton, we talked about that. We talked about central, cilium flagella. I think that's it, unless you want to do the review questions at the end?
No.
Oh, okay, if you want to do them, you can. And then I will pause this video and then stop it once you have done it. You can go in as any name you want. Okay, so everybody had a chance to do their questions. Let's see. Yay! So I'll read it out loud in case you're watching this home and can't see it. Which organelle is the powerhouse? That would be the
Mitochondria.
Mitochondria. And I'm a highlighter. Uno momento. Determines what goes in and out. Cell membrane. Somebody missed that. So sad. Processes, packages, and distributes is the Golgi apparatus. Stomach is the lysosome. Protects and is permeable to gases and water so they can enter. So you want to keep in mind the cell wall has to be permeable because things have to go through it. But a cell membrane you would refer to as being semi-permeable. Yeah, so good. Cell wall. Wait, did we get the last one was 100? That's 3. Okay, so we have 3 in a row. 3. Okay. 4. Control center is the nucleus. Good job. 5. Compartmentalizes the cell. Rough if it has ribosomes is the ER. Light energy into carbohydrates. That's number 6. Chloroplast. Another, number 7, multi-purpose organelle are the vacuoles. 8, protein synthesis, the ribosomes. Oh, okay, 8. Good job, you guys. Found in both plant and animal cells. Mitochondria are found in both plant and animal cells. 9 sets of 3 microtubules are the centrioles and the basal bodies, and what do basal bodies do? Anchor what? Cilia and flagella. Cilia and flagella, that's what basal bodies anchor. And number 13, 9 sets of 2 are cilia and flagella. Okay, any other questions or concerns about this chapter you want me to go over? Okay, you're super smart. Good job, I'm already proud of you.
Love you all, let's go!
Love you all.